Vai voltar para aqui a tela, né? Ok. Bom dia a todos e todas. Meu nome é Leonardo Farias e estamos reunidos para mais uma sessão científica do IGM, que ocorre todas as sextas-feiras, às 9 horas. Gostaria de lembrar a todos que essa sessão está sendo transmitida e ficará disponível nos canais do YouTube. Para mais uma sessão científica do IGM, que ocorre todas as sextas-feiras, às 9 horas. Gostaria de lembrar a todos que essa sessão está sendo transmitida e ficará disponível nos Pronto. Estará disponível nos canais do YouTube a partir da próxima semana. Hoje nós temos o prazer de receber o professor Kaye. Vamos mudar para o inglês. Good morning, everyone. I'm Leonardo Farias, and I'm here to another IGM scientific session, which takes place every Friday at 9 a.m. Today, our speaker is joining us remotely. I'd like to remind you that this session is being streamed and will remain available on IGM's YouTube channels. Today, I'm really happy to welcome Professor Kaye. I thought it would be nice to have someone more closely connected to leishmaniasis field to introduce him. Since Professor Washington, our advisor director of research, is away at an event representing Fiocruz, but send his regards to Paul. I have asked Patricia Veras to introduce you. So, Patricia, please.
Hi, Paul. How are you?
I'm good, thank you.
I'm very glad to be here. I'd like to thank Leonardo, for inviting me and giving me this opportunity to introduce you here at Fiocruz, Bahia. You know that you are missing us. You have to come back. We have to organize your presentation here in our institute again. Paul Kaye, I think this presentation is mostly for the youngest students, because Paul Kaye is a leading immunologist at the University of York. In my point of view, he is one of the most prominent researcher in leishmaniasis field. And he is internationally recognized for his several years, I will not put the number of years, of research on the immunology and immunopathology of leishmaniasis. His work focuses on host-parasite interaction, mainly in the roles of macrophages and dendritic cells, and explores the biology of inflammation and inflammatory response of host cells. And in this field, he does the thing very well, is to correlate the results in basic science to translate it in the possibility to develop chemotherapeutic interventions for visceral leishmaniasis. In this field, he has several collaborations. He has a phase 2 study for vaccine in other countries. And I think that in my point of view, we have today an excellent presentation and a very good research and a very nice person to discuss thereafter. Thank you very much, Paul, for to accepting our invitation. Okay.
Thanks very much for the introduction and for the invitation to speak. It's a great pleasure to be able to talk to my colleagues and new students in Brazil without having to spend 2 days on a plane to get to you. I do miss the caipirinhas, however, so that's something that we can't get around if we do everything by Zoom. Okay, so probably for the last year or two, I've been spending most of my time when I give presentations talking about human immunology, human immunopathology, and in particular, the work we've been doing trying to understand how immune checkpoint molecules regulate human immunopathology, particularly in cutaneous leishmaniasis, but also a little bit in visceral leishmaniasis. But I thought today I would talk about something very different and introduce a very different type of experimental methodology that a few of you may be familiar with, but I would suspect many of you are not familiar with. And that is the use of population dynamics to try and understand parasite dissemination in murine models of visceral leishmaniasis. So I thought I would start with a few slides just to introduce leishmania, and I won't dwell on that because I'm sure many of you are familiar, but there may be some in the audience less familiar. And so, oops, let's just get the slides to move. Okay, so just to remind anyone who doesn't know anything about leishmaniasis, this is a sandfly transmitted protozoan infection where parasites are transmitted into the skin during sandfly bite. They are intracellular parasites in the mammalian host, so they are rapidly phagocytosed by a whole range of different myeloid cells, predominantly monocytes and macrophages, as well as neutrophils and dendritic cells. And then the course of infection subsequent to that can be split very simply into two main groups. There's visceral leishmaniasis, where parasites leave the skin, disseminate to the visceral organs, most notably the spleen, the bone marrow, and the liver. And that gives rise to the systemic disease Kala-Azar or visceral leishmaniasis. On the other hand, some species of parasites stay localized within the skin, but immune priming to antigens derived from those parasites takes place in the lymph node, and then activated lymphocytes are recruited back to the skin to cause the various manifestations of cutaneous leishmaniasis, and you can see many of those shown pictorially in the top there. And of course, there are combinations of these forms. We know that mucocutaneous leishmaniasis is a metastatic disease originating from a single ulcer elsewhere in the body. And we know that post-Kala-Azar dermal leishmaniasis originates from visceral leishmaniasis. So there is some evidence, that even directly looking at these images, that dissemination may be occurring in different ways during this disease. In terms of the impact of leishmaniasis, I just stole this slide from the WHO website. This is their infographic. I think many of you will know this. About 1 billion people still at risk. It's a poverty-related disease. Mucocutaneous disease, highest prevalence, around up to 1 million cases per year. And I think we're starting to understand more about the impact of stigma and social exclusion, as opposed to just the impact of primary disease with cutaneous leishmaniasis. Visceral leishmaniasis, again, still high mortality rates, even though there's been fantastic success in eliminating Kala-Azar in the Asian continent. Currently, most of our prevention and control strategies are historic. They involve vector control, they involve diagnosis and surveillance, and are very difficult to implement in many settings. This is, I put at the bottom here, apologies to the immunologists, because I always suspect there may be some non-immunologists in the audience. So just a very, very quick summary of the immunology. We know this is an intracellular pathogen. We know interferon gamma is a major mediator of host protection, and that is principally derived from a variety of T lymphocyte subsets, as well as perhaps by natural killer cells. We know that macrophages themselves have different levels of innate control over Leishmania parasites, and this may vary between different parasite species. And we also know that interferon-dependent immune responses can occasionally be exaggerated, can lead to overt local skin pathology, as can the activation of effector cytolytic cells, whether they be CD8 T cells or natural killer cells. And so really the balance of pathology versus protection is mediated by a whole range of immunoregulatory mechanisms, of which perhaps IL-10 production, again by T cells and macrophages, is one of the best characterized. And I'm afraid that's pretty much all I'm going to say about the immunology. So anyone who wanted me to list 20 different CD numbers or 15 different cytokines, I'm afraid you're going to be disappointed from now on. So the final bit of introduction is just where we are with new control measures. In terms of the drug arm, the DNDi has been working, it's the Drugs for Neglected Diseases initiative, has been working very hard to improve therapeutic options for both visceral leishmaniasis and cutaneous leishmaniasis. You can see on the left-hand side here, there is a whole range of clinical trials that are either just being completed or are ongoing. But most of those use pre-existing drugs that have been around for many years, and the aim is to identify new appropriate combinations that either reduce severity of side effects or increase compliance. The one exception to that is at the bottom there, Lexi408, which is an orally available proteasome inhibitor, which is currently in clinical trial in Africa and India, and that's in phase 2 studies, and that's really the only novel compound that's currently in clinical trial. Vaccines have been challenging to develop. This cartoon just shows you the pipeline that we have for vaccines that are near or in the clinic. It's very sparse, as you can see. We conducted a phase 2 therapeutic trial in Sudan in post-calorazole dermal leishmaniasis. Unfortunately, that failed to have clinical efficacy. That was using a chimpanzee adenovirus vector. There is a Leishmania centrin knockout parasite, a genetically attenuated parasite, being developed by Geneva with collaborations with the FDA and others in the US. And that is hopefully going to enter phase 1 later this year. And there is also discussions around the development of RNA-based vaccines, particularly with the group of Steve Reed and others at HDD Bio. And in fact, we're starting a collaboration with them, hopefully, to introduce part of our antigen constructs into their RNA vaccines. But it's a very sparse pipeline, and nothing really has yet emerged to allow us to be hopeful that there will be vaccines in the next 5 to 6 years at least. Excuse me. So that's where I'm going to stop about the introduction. And I'm going to go on to this question of dissemination. And sometimes we think in science that we've always come up with a bright new idea or another reason to study something. Dissemination has been a question that's been on people's minds for many, many years. And I just show you this quote here from Sir Patrick Manson back in 1905. So really right at the beginning almost of the field of tropical medicine, where he talks about the occurrence of parasites, these Leishmania parasites, and how they are being transmitted to sandflies. When you have most of these parasites actually, particularly in the case of visceral leishmaniasis, held within systemic organs. So it's not a new question. And I think that's important for all the students to recognize that sometimes if you go back in history, you'll find the technologies are very different, but the questions being asked are very similar. And perhaps it's sobering to think that we may have not answered some of these questions that have been around for over 100 years. So I'm going to spend the next few minutes just reviewing a little bit about what we know about dissemination, or what we think we know might contribute to dissemination, before going on to this new experimental approach of population dynamics to try and understand a little bit more about how dissemination actually occurs. So firstly, why is it important to understand dissemination? Well, clearly, as I've already shown you in that first image, it's intimately linked with disease pathogenesis. And our entire classification of the different leishmaniases into visceral, mucosal, and other forms of disease really reflects that dissemination is occurring in some way or another. Secondly, it's important, as Patrick Manson identified to understand dissemination in the context of transmission, particularly again, in visceral leishmaniasis, where there is no obvious lesion, which may be the source of parasites for a feeding fly. There are also very practical implications for understanding dissemination. When we treat clinically with drugs, most of our understanding of the pharmacokinetics and the pharmacodynamics, i.e. the distribution of the drug and its ability to kill parasites in different tissues, is based on experimental models and experimental analysis of drug action in the liver and spleen. And that's true even in the clinic for visceral leishmaniasis, where effectively clinical cure is defined by changes in systemic parasite burden. What we know very little about is how drugs might operate in different sites if those sites also are parasitized. And that becomes very important then when you start to think about why do we have relapse, why do we have post-calorectal dermal leishmaniasis. We know that's partly to do with tissue-specific immune control, but it also relates to the fact that those drugs may not be operating effectively in those areas. So I think there are a number of reasons that I've just mentioned which make this a very important topic to consider. So what's the evidence then for dissemination? If you look in animal models of disease and with high-resolution imaging techniques, it's now clear that besides the classical sites of parasitism in visceral leishmaniasis of spleen, bone marrow, and liver, there are multiple cryptic sites of parasitism. You can see in the images on the right-hand side, parasites can be found in the brain, parasites can be found in the thymus. Much of this work done by Thierry Lang and colleagues at the Pasteur Institute, as well as many others. We call them cryptic sites of parasitism because most of these sites actually do not show overt clinical pathology, even though parasites are clearly present and identifiable. It's a whole other topic as to why they are not showing sites of overt pathology. Secondly, if you look in parasites with HIV-VL co-infections, they again have a far greater extensive pathology than would normally be recognized in non-co-infected individuals. So, for example, there is involvement of the duodenum, the stomach, the skin, etc., in multiple patients. And with the related parasite to Leishmania donovani, Leishmania infantum in dogs during canine visceral leishmaniasis, we can see this really extended parasite tropism to different tissues. And then finally, a study from Ethiopia conducted a few years back using these microbiopsy punches was able to show that parasite DNA could be found in the skin, even of healthy controls in endemic areas, as well as in asymptomatics. And it really was not associated with any obvious sites of sandfly bite, if you like. So I think there's a lot of circumstantial data that's out there that shows parasites are not just where we've always thought they accumulated in the main. The second bit of evidence comes from trying to think about parasites as intracellular parasites. In other words, their dissemination is largely dictated by the potential movement of their host cells. And what we know about Leishmania parasites is that they are particularly good at parasitizing migratory cells. And this is just a cartoon from a recent study from Abhay Sataskar's lab using dual RNA-seq to identify which cells have parasites. And you can see on the left-hand side here, not just conventional macrophages and monocytes, but a whole range of other migratory cell populations can be shown to contain parasites. Neutrophils are not shown here because this is done on peripheral blood mononuclear cells, splenic mononuclear cells. What they were also able to show in the same study, and has been shown many, many times in vitro by infecting macrophages, for example, with parasites, and on the right-hand side you can see an example of this, is that infected cells have a very different transcriptional profile from uninfected cells, hinting at the possibility that infection may alter the migratory properties of these cells. although that's been very poorly demonstrated either in vitro or in vivo to date. The third bit of evidence is related particularly to skin parasitism, where we've spent a lot of time trying to understand how skin becomes parasitized during experimental visceral leishmaniasis infection, where in fact the route of infection is intravenous and therefore bypasses the skin completely in the initiation phase of disease. And what we were able to show in a couple of manuscripts was using an ecological mapping approach that you can see at the top here. We were able to show that parasites actually are found dispersed across the skin, but not in a uniform manner. In fact, they are dispersed in a very patchy manner, or an over-dispersed manner, where certain areas of the skin have these patches of parasite accumulation, and other areas of skin are completely unparasitized. And we were able to use computational and mathematical modeling approaches based on various ecological principles to come up with a schematic that you can see that's just playing in the bottom here of how these patches might develop from an initiating infected monocyte entering the tissue to lateral spread of parasites within the tissue to generate patches that are of different size and different composition. And through modeling and xenodiagnosis experiments, we were able to show that the presence of these patches actually facilitates transmission to sandflies. And then the final piece of evidence I just want to show you is that if one's thinking about cryptic sites of infection, one should not forget that cryptic sites of infection can also vary in their immune status as a consequence of systemic impacts on immune function. And so we've spent a lot of time over the last few years, and again, I'm not going to talk about it in any detail, using spatial profiling techniques to look both at the primary sites of infection, for example, in the liver, as shown in the top here, where we've been combining lipidomics and transcriptomics to map lipid biochemical changes within granulomas. But more importantly, we've been looking at these cryptic sites, such as the skin, to see how the landscape is reshaped by the presence of systemic infection. And what you can hopefully see in this cartoon here, this is a Visium spatial plot, just delineating various substructures within the skin. And in these heat maps on the right-hand side, you can see the first two columns are healthy skin, and the following three columns are infected, or skin from infected animals, although minimally infected skin. And you can see that every one of these different spatial compartments changes in the regulation of immune-related genes as a consequence of systemic infection. And this is something which has been well described in a whole range of other systemic diseases that, as you might think, the immune responses spread and modify alternate tissues. So that's 4 sets of data that relate to dissemination, but they don't actually tell us the answers to some very, very important questions. They don't tell us when colonization starts to occur and in what way is that related to systemic disease or immune changes, perhaps immunodeficiencies associated with those systemic changes. They don't tell us where disseminating parasites actually originate from. And they also don't tell us about the mechanism, the actual mechanisms of dissemination in terms of which host cells. So we have a lot of circumstantial evidence on how. We have a lot of sort of data about when that's derived from clinical disease and from models. But the really difficult one to address is the where. Where do disseminating parasites originate from in terms of other tissues within the host? And this is something which you really can't address by single snapshot studies of individual animals with the conventional techniques that we've been using before. So the way we decided to go about this was to take a lesson from our bacteriology colleagues who've been using for a number of years population dynamics to study dissemination. So what do I mean by population dynamics? This is the, if you like, the prototypic paper here on sequence tag-based analysis. And the aim here is to generate parasite populations where individuals within that population have unique barcodes, which serve, if you like, as alleles that can be monitored and distinguished between individuals. And the idea is that you can then take the whole population and compare the population compositions between different sites, and with a whole range of mathematical approaches, start to infer things about the dynamics of spread and even the direction of spread of parasite populations or pathogen populations. And although this has been used quite extensively in bacteria, there's really only one study so far, and that's from Matthew Child's lab, applying this to a parasitic disease, in that case, Toxoplasma gondii. And that was really being used to try and understand a little bit about so-called bottlenecks to infection. So let me just describe what type of data we can get from these studies. And I'm going to keep this fairly light touch because I don't want to go into the mathematics at all. But if you consider that we have a parasite population now, and each of these colors represents a unique barcode that allows us to identify the composition, population composition. We can measure something called a bottleneck size. In other words, what is the pressure on these parasites as they go into different tissues to be able to colonize. I'm not going to talk very much about founder populations today. We can measure diversity, how similar are all the parasites within a particular target tissue, and then this is measured by something called the Shannon index, which is a measure of so-called alpha diversity. We can compare individual animals by comparing their total body populations of parasites. But the one I'm going to spend most time on today is this metric here called genetic distance, where we can computationally derive a value for how close, how similar are two populations of parasites in two different tissues within the same animal. And from that information, we can use graph theory approaches to start to understand directionality of movement, perhaps, between those different organs. So I'll come back to some of these concepts in a minute when I show you some more data. So the general schematic that we've been studying, and this is really pioneering work done by a PhD student, Chiara Loffrey, in the lab. And what she did was to make a barcoded library of Leishmania donovani. I'll show you how that was done in a second. What that generates then is a population of Leishmania parasites with individual sequence tags in them, which allow them to be new. And very importantly, these have to be neutral. So in other words, they must not affect function. We can then infect our standard experimental model of visceral leishmaniasis, which is the B6 mouse. Take out any organ of interest, count and identify each of the barcodes using Illumina sequencing, and then apply a whole range of mathematical approaches to identify how similar and how different those populations are within those tissues. So to take you through a little bit of the data, and this is all unpublished data as yet. Firstly, with the help of Jeremy Mottram and his colleagues, also at York, we generated a barcoded library, or Chiara generated a barcoded library. And this was small 20 base pair barcodes inserted into the ribosomal locus of Leishmania donovani. An important consideration is how many different individual barcoded lines do you actually need to be able to generate good mathematically robust data. And so this cartoon down the bottom here, I won't go into great detail, is a computational simulation to try and understand exactly how many barcode lines you need to be able to generate appropriate data. And it turns out that you need about 100 or so as a minimum. And so Chiara generated 100 individual Leishmania donovani lines, and this is 100 separate transfections to generate these. These are then pulled back together again when we do the barcoded line infections. I mentioned it's important to make sure that these parasites are isogenic and have the barcode is of neutral function. We anticipate that from entry into the ribosomal locus, but it's important to test it. So it's tested by comparing in vitro parasite growth rates, and these are all statistically identical, by comparing macrophage infectivity, and I'm just showing you 5 of the lines here. Again, the frequency and abundance of infections within macrophages in vitro is the same, and the kinetics of survival over the first few hours in macrophages is also the same. So we're fairly confident that this barcoded library does not have any biases because the barcodes are affecting certain functions. So this is just a graphical representation of the sorts of data that you then get out of these experiments, which looks very complicated, and it is a little bit complicated, and it certainly takes a little bit of time to get your head around this. But what we have is an input pool of parasites, and this is a mixture of these 102 different lines. We know what we put in terms of the frequency. We can then wait different periods of time, and the data I'm going to show you is either early in infection, around 4 weeks, or in the chronic phase of infection in the mouse model, which is around 10 to 12 weeks. You can excise various organs, do the sequencing, and what you can see is that some organs have lots of parasites, some have few, and you can compare the levels of diversity across these different tissues by various formulae. So that's what the data looks like, and you do this for multiple individual mice at multiple time points as you wish. And we've generally used groups of somewhere between 5 and 12 animals at each time point to be able to get robust data. So let's just look at some of the data and some of the conclusions that we're starting to get from these studies. And I just want to re-emphasize that really this type of approach has never been done before in the context of leishmaniasis, and is something which you can't infer from any other type of experimental approach. So if we just look first of all at the liver as an exemplar of this data, and I'll come back to other bits later on. If you look early versus chronic infection, and this is the Shannon index. This is the index of diversity of parasites within that particular target tissue. So there's a fairly homogeneous mix of parasites, and each of these dots is an individual animal in this case. Early in infection, there is reasonable homogeneity and relatively low level of diversity across liver parasite populations. But as you move into the chronic phase of infection, you start to see an increase in diversity. And that increase in diversity would not be due to selective pressure on one line or another, because these barcodes do not interfere with the biology of the parasite. And it's also not due to selective expansion of particular codes. And so the only interpretation for this really is that colonization of the liver actually occurs multiple times during the course of infection, leading to an increase in diversity. Now, clearly, we've only infected the mouse once, so the assumption must be, therefore, that if one's having continuous colonization, that colonization must be appearing from other sites. And you see the same thing to a certain extent in the bone marrow, and you can see quite a bit of heterogeneity in the bone marrow. In the gut, in the lymph node, and in the lung, parasite numbers are actually much lower, and that contributes to some of the scatter in the data. But it also suggests that these particular sites have a different route of colonization from that we see in the liver and the bone marrow. And that perhaps is not surprising, because we know the liver and the bone marrow and the spleen to some extent are the primary RES organs that will clear parasites early during intravenous infection. So this starts to give us a little bit of an idea about the diversity of parasites in different sites, but it's not telling us anything about why. Before I move on to the why, I should just talk a little bit more about the skin, because I'm going to keep coming back to the skin. It's been an area that's particularly interested to us. We first wanted to show that in the model we were currently using, this patchy distribution of parasites in the skin also occurs, and this is just showing similar images to the ones I showed before, but using a TD tomato variant of Leishmania donovani. And this is just an image of the skin. You can see a big patch here, another patch here, smaller patches. And so there are areas of completely uninfected skin. So there is diversity in the skin in this patchy way. When you look at parasite skin diversity between early and chronically infected animals, there's actually very little difference in the median. But the important thing to recognize here is that each of these columns is an individual mouse. And each of these biopsies, each of these symbols is an individual sampling, an individual biopsy from that mouse. So in other words, one sample might be here, another sample might be here, another sample might be here. We don't know exactly where they are because we can't prejudge where we actually take the biopsies. But what this tells us is that there is within mouse variation in the skin in terms of the diversity of parasites. And just to illustrate that without formally linking the data, one could imagine that this patch is a large patch and has very diverse populations of parasites, whereas some of these smaller patches may be much more homogeneous, suggesting that seeding events may be different, that this might be early seeding with a low diverse population of parasites compared to this patch, which may have grown and recruited more parasites from different sites. And you can see individual mice are quite different in the degree of scatter that they have. So we think we have essentially inter-patch diversity, which is likely stochastic, but also represents this independent seeding of different patches. So if there is independent seeding, what sort of relationships can we start to draw between parasite populations in different tissues? And this brings us on to this concept of genetic distance. So just to remind you again, genetic distance is the comparison of the composition of the parasite populations in two different sites. If they're very similar, the parasite population, then the genetic distance is small. If they're very different from each other, then you have a large genetic distance. And we can make comparisons pairwise across all of the sampled sites that we've looked at in a particular animal. And I should say again, just stress, this is done on an individual animal basis. And so in this heat map, you can see a comparison and genetic distance here. So small genetic distances are in purple, and the lighter the coloration, then the more dissimilar those populations are from each other. And so this individual mouse just shows us some interesting things. It shows us that all of the skin biopsies we took from that mouse are very different from most of the systemic samples. So this is the skin biopsies compared to liver, lymph node, lung, and spleen. But all of those skin biopsies are actually extremely similar to the bone marrow. If we take a different example, look at the liver, and the population of parasites in the liver of this particular animal are very similar to the populations we find in the lung and the spleen, but quite dissimilar from the skin or the bone marrow. So you can build up these pictures for each individual animal and turn those into a connectivity plot. And this animal in the heat map is just shown in the bottom left here. And you can see the connectivity of this animal. This is the bone marrow here. It's highly connected to skin, and each skin site is highly connected to each other. But essentially the viscera in this particular animal, besides the bone marrow, is relatively independent in terms of its population structure. Whereas if we switch to a different animal, you see the reverse. You see in this particular animal, the viscera is highly connected and the skin has very low levels of parasitism and is actually very poorly connected to the viscera, with a few exceptions. So that's good. It tells us that a little bit about connectivity, but it doesn't tell us anything about directionality. It doesn't tell us whether one organ is seeding another organ or vice versa. In other words, what is the flux, what is the movement of parasite populations that gives rise to this stable structure that we see here? And the way you address that is to use another set of metrics. I'm not going to go into detail again, but effectively, experimentally, we dissect the mouse, we take all the tissues out, we filter to remove things that will generate noise. And then what we do is compare the population structure between two different organs in both directions. So you're comparing A versus B and B versus A. And you can then generate from that a heat map, again, another heat map just to make life easy, to identify which organs might be contributing to the population structure in a different organ. Maybe that will be a bit clearer when we just look at the data here in this heat map. What it shows us is this axis here is our source tissue, and on the bottom here is our recipient tissue. And what it tells us is that these two particular skin sites, number 9 and number 1, seed parasites. They have an increased ability, their parasite population structure is much more similar to many other sites, not just in the skin, but also in the viscera. So these are what we might call superseders. They're sites in the skin where their parasite population has been spread out to accompany other tissues. If you look at the other way around, in contrast, the spleen is, if you like, a promiscuous recipient. It receives parasites from pretty much all of these different sites. But actually, the spleen itself is not very good at spreading parasites around the body in this particular mouse. And if we use various models of network theory, and again, I won't go into the details of these, we can start to represent this graphically, as you can see on the right-hand side, with directionality in terms of these arrows. So in this instance, this is what Chiara calls the parasite highway for this particular mouse, where parasites move predominantly from skin site 0 to skin site 4 to skin site 1, and then from 1 elsewhere. And so this is their trajectory of movement within the skin predominantly, with a little bit of movement out to the lung and a little bit of movement out to other systemic tissues. In a different mouse, this goes back to the one I showed you before, this is very much a systemic orientated mouse where most of the movement is between bone marrow, liver, and other visceral tissues. So the conclusions really from this are that, and I think really something I didn't predict when we were doing these experiments, I had in my mind that spreading from the viscera would be a one-way traffic, and you would traffic from the viscera to the skin, and those skin patches would remain as skin patches of parasites. I think what this data shows for the first time is that the skin, in fact, can seed back out to the viscera. And that, I think, is going to be important for thinking longer term in a translational way. It shows us that different organs can be promiscuous, and it shows us that different highways exist, and different mice have different behaviors. So the final thing I want to show you in the last couple of minutes is just to address the question of whether these highways are actually stable within an individual mouse. It's hard to do that within an individual mouse because you can't sample the same mouse twice for obvious reasons, that it can't give you its spleen or its liver twice. So unfortunately, we have to pull the data for this. But the experimental approach we took was to do a standard chronic infection using our barcode line, but then to put an immune challenge back into the animal by way of a secondary infection. And in this case, we used luciferase infection parasites so that we could show that there was indeed a protective immune response generated by that secondary infection. And then you can do very similar analysis to what I've already shown you and ask, how does that secondary infection alter skin-to-skin seeding or skin-to-visceral seeding? So what was surprising in this data is when we look at the skin, this is the data for the single infections, and what you can see, a very highly significant reduction in both the diversity as well as the number of parasites in the skin. And I think that's indicative of protective immunity, as one might have predicted. What we didn't predict, however, was that when you look at the connectivity from the skin to the viscera following a secondary challenge, there is actually an increase in migration. This is a log scale, so it's a little bit squished, but you can see very high p-values here. There is an increase in migration from the skin to the viscera. So I think this tells us a couple of very important things. Firstly, it tells us that you can change, you can modify these pathways, these highways, if there is an appropriate stimulus. Now, we've used an immunological stimulus in the sense of reinfection, but it's not clear at this stage whether it needs to be an immunological stimulus or whether it could be any other form of perturbation, for example, change in diet, change in microbiome, change in other aspects of co-infection, for example. But it suggests that we can move these parasites around in different directions by manipulating the system. So I'm just going to finish then with a few conclusions. And again, I'm pretty sure this is a very different way of looking at things compared to what many of you may be used to. So firstly, I think we've shown with this approach that parasite dissemination is dynamic and it can be influenced by environmental changes, including reinfection. I think we've shown now that the skin and the viscera are interconnected, and we probably knew that from static analyses, but what we didn't expect was that the exchange was going to be in necessarily both directions. And we've also identified that some skin patches, and we don't know their unique characteristics yet, but some skin patches seem to be able to be super spreader patches. As you can see in the cartoon here, where parasites are seeding potentially multiple migratory cells, which then enter the circulation and go back into the viscera. So what we're doing currently, and what we're hoping to be able to address over the next couple of years, is to confirm which vehicles, if you like, are trafficking down these pathways. And I think many of us would assume that those are the myeloid cells that are the predominant populations affected by Leishmania donovani. We don't really know what determines this roadmap. We've got a lot of circumstantial data, as I said earlier, from high resolution deep phenotyping studies, but trying to link that exactly to these pathways is still open. We don't know what drives inter-mouse variation, and I would suspect if you extrapolated this to humans, you would see the same level of inter-human variation. I'm not sure that we will necessarily all be able to answer that precisely, because it's likely to be involved with all aspects of the animal's biology. And then finally, the consequences for drug and drug resistance and relapse are something which I think hopefully are fairly apparent. If our drugs do not kill all our parasites in the skin during treatment for visceral leishmaniasis, I think our data suggests that there is the capacity for the skin to reseed the viscera and allow for potential relapse. So on that note, I'll just stop, I think, and leave some time for questions, and just to acknowledge all the people that have done the work. Really, it's a monumental effort for her PhD by Chiara, who's really worked on this both doing the computational analysis as well as the experimental wet lab work. A lot of the background data from my lab has been generated by Nidhi Day and Shumit Day, who've been, and particularly in the human pathology space, with help from Helen Ashwin. A lot of help from Jeremy Motran and one of his postdocs, Juliana, for generating the CRISPR library. And John Pitchford, who's our resident mathematician, ecologist, who's really been a guiding force for telling us that immunologists shouldn't ignore ecology, and that how the immune system operates within an individual is not that different from how many ecological principles operate in the greater, wider field. So I think, obviously, funders, mainly the Wellcome Trust, for this particular work. So I'm going to stop there and then hopefully take questions. Thank you.
Thanks, Paul. Amazing talk. It's great to see how technology can be applied to answer biological questions. So I think I'm open. Questions here in the audience. Patricia.
Oh, that's fantastic. Big results, and actually opens so many questions that we don't know how to begin but my question is related to the super spread patches.
Mm-hmm.
You anyway explain why do you have this formation of super spread patches, and they can disseminate, or they can migrate or disseminate wherever in different tissues, if well understood, but don't have actually a preference for any one of them. That's for sure. And so two questions. If you know how they are formed, and then why they go directly to all the tissues, but not have any preferences. I ask you most interested in the dissemination that occurs in cutaneous leishmaniasis, that we can go further and make I don't know, queries about using your results as model, and then go ahead.
Yeah, I mean, just to, I mean, the first thing to say is I can't fully answer the question. We don't know. But you know, that's the fun of biology, is doing things that lead you to things that you can't answer. The second point is I think it would be really fascinating in the you know, if one was extrapolating this to mucocutaneous disease, are super spreaders do super spreaders still exist, or are they super spreaders to a specific location, i.e. the mucosa, rather than broadly speaking? So one of the challenges with trying to address this experimentally is that there's no way to identify a particular patch in time or space and then follow what leaves that patch as to where it goes. So you just can't do that experiment for obvious reasons. So the way we're looking at that is to try and look at the heterogeneity of both the parasite and the infected monocyte macrophage populations within different patches. And so there are, and some of you may have noticed the subtlety when I showed you the TD tomato image. Some of those patches are very bright, some of those patches are very dull. And what we know about TD tomato expression is that it varies depending on the metabolic activity of the pathogen, of the parasite. And so there's a nice study from the lab in Belgium looking at quiescence markers, and in fact they used TD tomato as a surrogate for quiescence. So you could extrapolate, and this is really wildly extrapolating, that the patches which have metabolically high activity parasites are the super spreaders, and the parasites with metabolically low, potentially quiescent parasites maybe are not the super spreaders. But I can't prove that in any definitive way. So we're doing a lot of spatial mapping, trying to map parasite transcripts as well as host transcripts in situ in individual patches to see whether there is a heterogeneity. And you could imagine that the microenvironments in those patches would be slightly different as well. So I think that's a bit of hand waving, but immunologists are good at hand waving, as you know. So.
Question more? Or do you have another one? I can't hold it. It's about the super.
Let's
Go.
intervene with the chat. Okay. So Natalia asked basically the same question for talk. Do you plan to characterize those skin pads that are super spreaded, and if so, how? So it's already addressed it. And Claudio, congratulations on your excellent work. It demonstrates that the full complexity of the disease and the need for similarly complex approach to study. Just a comment. Professor Washington Corrado say, good morning. It's an honor to have Dr. Poe with us. And I think that Mika had a question, or look at that.
Good morning, Professor. Thank you very much for your very nice talk. That's interesting. Different from leishmaniasis, leptospirosis, when we have infection, the leptospirals disseminate through all the organs. But in the very end, they just show up in the kidney.
Mm-hmm.
Really interesting. My question is, from what I saw, you test this using female mice. Is that true? Only female. Have you tried in male and see if you have the same result?
We have tried some of these experiments in males. One of the difficulty with male mice, if you've studied male mice, is that they love fighting each other. Yeah. And what you end up with is a large number of secondary skin challenges through bites and various other things. So because we had a particular interest in the skin, we decided to use females only for most of these studies. I think it's quite likely that there would be sex differentiation differences as well between migratory routes.
How about to use the male mice, but keep them separated in one package? We have the same results.
Unfortunately, our regulators will not allow us to keep males as separate mice. So yeah, we can't do that experiment, I'm afraid.
Thank you.
Another question here in the audience. Patricia, another one?
Yes. I'm also curious, Paul, if you did that, I don't know, I didn't actually saw you showing this result, but did you try to infect in vitro those guys and found that the cells respond differently to the population of the super spread, all them mixed or individually, to know if the macrophages respond differently to the infection regarding the nature of the parasites, genetical nature of the parasite?
Yeah, no, I think we've done a lot of those experiments, and the whole basis of the approach is that the parasites, even though they have these barcodes, have no difference in their biology.
Okay.
Okay, so the barcode has to be neutral.
Okay.
Otherwise, you would have selection pressures which would change the population dynamic. So these are all neutral, and so we've done all of our in vitro experiments show that really, if you compare all these different barcoded lines, they all behave the same in macrophages. So the technique can be used in a different way by introducing barcodes into virulence factors, for example, to understand what's a virulence factor and what's not a virulence factor. But that's a very different approach from the one that we've taken, where neutrality is important.
Okay, Juliana.
Hi, Paul.
Hi there.
Congratulations, it's like really amazing work. I was just wondering, like, based on what you found, and you're probably gonna say you don't know, but I'm still gonna based on what you found for visceral leishmaniasis, what do you think it happens in patients, for example, with a disseminated form of the disease or the cutaneous disease? caused by leishmaniasis. Do you have any idea?
I suspect that, I mean, I think there are two things that are important, and it's true in disseminated CL, but it's probably also true for PKDL as well. Firstly, I think the dissemination is likely occurring stochastically over time from those visceral sites, and that's sort of what's supported by the data we have here. But I think the important thing with both PKDL and possibly with DCL as well is that it also requires the immune response to focus itself on those specific skin sites to be able to see disease. And I think perhaps in the context of PKDL, a misinterpretation that you often see in reviews and in textbooks is that after you treat for VL, the parasites migrate to the skin. I don't think that's the case. I think the parasites are always in the skin. They're always being seeded, and there is this migration that I've been talking about. But the critical determinant is whether the immune response switches from a cryptic site to an active site. And that may occur differently in different tissue sites. You know, we know with PKDL in Africa, for example, it always starts on the face and then moves down to the trunk. I find it very hard to conceive of parasite dissemination starting with dissemination to the skin in the face and then disseminating somewhere else. I find it much easier to see a picture where the immune activation is more easily facilitated in the face because of UV exposure, etc., and then migrates elsewhere. I don't know whether that might occur with diffuse disease as well, where not all lesions erupt at the same time. It's progressive, but it may not be all to do with dissemination, I guess yeah.
Thank you very much.
I have one question, Paul. Is actually it's amazing how do you produce the parasites, the isogenic barcode parasites. But I was wondering if you observe hybrids or some cross exchange of the genetic material between the isogenic barcodes or during your analysis. And the second question is if you plan to analyze the transcriptome of these parasites from different sites and see if they are expressing different genes that can explain the different sites, etc.
Yeah. So the first question, the answer is no, we haven't looked. I suspect there might be some exchange, but it's probably not influencing our data too much. In terms of the parasite transcriptome, we've been doing, we have preliminary data now using cosmic single cell spatial analysis. And what we've developed is a 100 gene parasite panel for use in the Cosmics platform. So we can't do the whole transcript in Leishmania, but we have 100 marker genes related to various key functions that hopefully we will be able to do dual transcriptomics spatially within individual patches. But that's probably a year or so down the road before we have that data.
Okay. That's a talk for another talk.
Another talk.
Any questions? I think that we are done. So I really would like to thank you, Paul, for accepting our invitation. It was a great opportunity to see how you are integrating different technologies and how you design the experiments, the assay, and address the questions. It was really amazing. So thank you very much.
Thank you for the offer. Great to see some old friendly faces out there as well.
Bye.
Bye.
Bye-bye.
Bye.